presentation on different aspects of RNA structure and biology. And so we start with Jean-Pierre Glouzon from the Proceedings Presentation. And we'll talk about, so this is a proceedings presentation. So we will talk about aliFreeFold.
Thank you very much for the opportunity that you gave me to present my work. So I will talk about aliFreeFold, which is an alignment-free approach to predict secondary structure of homologous non-coding RNA sequences. This work has been done by my advisor Aida Wangrawa and I, Jean-Pierre, at the University of Sherbrooke. I will first start by giving you the motivation why we decided to work on aliFreeFold. Then I will explain the detail behind the approach and the 5 main results that we got from our experiments. And take-home message. The prediction of conserved structure is still relevant. It is really important because it helps us to understand non-coding RNA functions. Why? Simply because there is a tight relationship between structure conservation and RNA function, and also from the fact that a vast majority of non-coding RNA families have a conserved structure. So here I put an example of a typical conserved structure of tRNA with a specific cloverleaf shape. Currently, we can see an increasing in the number of homologous sequences, but also in the number of families. So this indicates that there is also a growing in the conservation signals, because as we have more and more homologous sequences, we expect to have much stronger conservation signals. So we think that we need approaches that are able to scale with respect to the number of sequences, but also approaches that could potentially take advantage of the increasing in the conservation signals. With those 2 goals in mind, we decided to work on aliFreeFold. Basically, the idea is simple. The first step, the main idea of aliFreeFold is to find a way to efficiently explore the set of suboptimal structures coming from the homologous sequences, and from those suboptimal structures, identify the most conserved features and use those features to compute the best, what we think the most conserved one. Here I show you one simple example, 3 sequences from the hammerhead ribozyme family with the set here of suboptimal structures. As you can see here, there is the multi-loop is shared among the majority of those suboptimal structures. So we compute this information and we use this information to derive the representative structure. So basically this is the main idea of aliFreeFold. I will then get into some details. As input, we have a set of homologous sequences. We use in our framework RNA subop to generate the set of suboptimal structures. And using those suboptimal structures, for each of them, we compute a vector of occurring motif. So here, as an example, for the motif M, the multi-loop, we can see that the majority of the suboptimal structures have an occurrence of, the multi-loop occur once in the majority of those suboptimal structures. So basically, after computing the vector representation, all those vectors became the N-motif representation. Typically, an N-motif could be seen as a motif, like a multi-loop, a hairpin, a stem, or an eternal loop. But considering also the properties, for instance, a stem is usually associated with the number of base pairs. We also consider the neighbor of each motif. For instance, we'll consider a stem here with the stem here with the hairpin and the multi-loop, which are the neighbor of this stem. So here, as a result, we have the feature vectors from the suboptimal structure. So the next step will be to weight those features. Not all features are important. There are features that are well conserved and others that are not. The goal will be to design a weight function in order to give more importance to the features that are well conserved. So here, as you can see, the multi-loop here is well conserved because the occurrence here for the majority of the structures is 1. But it is not the same for the S bracket HM. There is some variability here. So to compute the weight for each features, we use an entropy-based weight function. So typically here, what we did is we would like to evaluate how, in a feature, for instance, how the distribution of the occurrence varies. So here, we just use this index is based on the diversity index, which in fact was used in ecology. And basically, this index gives information about in the distribution how many species or types we have in a data set. So typically, we use this entropy-based function in order to evaluate the features, the conservation of each features. After that, it's really straightforward to get to just by to multiply each in the n-motif representation, to multiply each features with the corresponding weight. Yes. So this gives us the weighted n-motif representation. Given the weighted n-motif representation, we compute the centroid, the mean vector, and we get just the structure which is the closest to the centroid. And this will be the representative structure using the Euclidean distance. So this is basically the detail for aliFreeFold. Then I will go with the experiment because I would like to see whether or not aliFreeFold is able to perform well. So we use the dataset of 30 families with some statistic here relative to the number of homologous sequences, the average sequence length. We decided to test aliFreeFold against the main type of approaches, meaning alignment-free approach, such as in aliFreeFold, align and fold approach, which typically compute the consensus structure from homologous sequences by aligning and folding at the same time. Align then fold. So the alignment step and the folding step are just separated. And the last type of approach, fold and align. So typically, those approach will compute the folding and then will align all the different structures in order to get the conserved one. To evaluate the predictions, we use the positive predictive value, PPV, the sensitivity, and the Mathieu correlation coefficient. The PPV gives us information about the average correct base pair found computed by each approaches. The sensitivity is the average number of true base pair found by each algorithms. And the Mathieu correlation coefficient, in fact, gives us a much more overall appreciation of the prediction. Why? Because it included the PPV in a sense and the sensitivity. So for the rest of the talk, I will mainly focus on the MCC here. So let's start with our 5 main results that we get from our experiments. The first result is the fact that considering the suboptimal structures can help greatly to improve prediction. Here, this graph, we report the average correct base pairs from the best suboptimal structures. Here, considering individual sequences, so I take one sequences, I generate all the suboptimal structures, and I pick the one which is closest to the validated one, and I report the average correct base pair. And I do the same thing using sets of 5 homologous sequences, 10, and more than 15 here. As a baseline, here I consider only the most stable structure. As you can see here, there is a clear increasing in the average correct base pair of the best suboptimal structures. And also the standard deviation significantly, by increasing the number of homologous sequences, decrease. So this indicates that there is something interesting in the set of suboptimal structure to consider. The second point is, as I mentioned, we compare aliFreeFold, as we know that there is interesting information around the suboptimal structure. So here we compare, using the MCC, aliFreeFold, and all the different type of approaches. So here, as you can see, aliFreeFold here and TurboFold are the top approach that we found performing well. So this also indicates that exploring the suboptimal structure can really help improving the prediction. So for the efficiency, we just recorded the running time against the average length of the set of homologous sequences that we have studied. And what we observe is, generally speaking, alignment-free approach are generally fast. Here, alignment-free approach are aliFreeFold here and RNA-cast here. It is important to mention that while centroid aliFold and aliFreeFold are close together, those two here are much faster. And the reason is as follows. Those approaches are aligned and fold. There is no simultaneously align and fold. So they are less demanding in terms of computation. This is mainly the reason they are the fastest. The reason why we only focus on the average length is because we observed a high correlation, statistically significant correlation of the running time against the average sequence length, but it was not the case when we consider the number of homologous sequences. As the fourth result, we were interested from the start to see how well all the different approaches are able to take advantage of the increasing of the number of homologous sequences. So what we did here in this experiment, we just increase gradually the number of homologous sequences and compute the MCC for each of those approaches. As you can see here, you can see that aliFreeFold is able to consider the increasing number of homologous sequences, which means he's able to consider the growing conservation signal. And the reason why aliFreeFold is able to do that, I think it's really because of its weight function. The weight function that we develop is based on the entropy. So with much more data, we expect to have a much precise score or a much relevant score, a much relevant entropy. So that's explain why, by increasing the number of homologous sequences, we get better results. And why also the weight function is, in fact, the main building block of aliFreeFold is basically from the fact that when we remove the weight function from aliFreeFold, the predictions start to get to be worse. Here, in red, we have aliFreeFold with the weight function, and here without the weight function here. So we clearly see a gap as the number of suboptimal structure increase here. So as a take-home message, we showed that studying the conservation of suboptimal structures can yield accurate prediction. And we try to use this knowledge to build an entropy-based weighting scheme in order to capture the increasing conservation signal coming from the growing number of homologous sequences. As future work, there is some improvement that could be made for aliFreeFold. The first one is aliFreeFold actually ignores completely the pseudoknot. We know that for some families, pseudoknot are really conserved. So our future work here is to use, for instance, VS subopt, which is an algorithm that that generates the suboptimal structures, but with the pseudoknot, in order to explore the conservation of pseudoknots. To explore the conservation of pseudoknots. The other point here is that some families have a portion of sequence that are really conserved. But so far, we totally ignore that. So here, what we are actually doing is to combine the sequence and the structural information. There is many ways to do that. And we take our inspiration from the multimodal machine learning approach, where we can see an RNA from two perspectives, the perspective of the sequence and the perspective of the structure. So the main idea will be to find the relationship between those two, in fact, spaces. The last point here is that aliFreeFold compute one representative structure. So what we would like to do is to not only have one representative, but for each sequence to get a structure. So the idea here will be to take the representative structure and to project this structure onto all the different homologous sequences in order to have, in fact, the set for each sequence, the appropriate structure. So I would like to thank the Cobius Lab, my advisor Aida, my colleagues, the University of Sherbrooke, where actually I'm doing my postdoc studies, and the funding agency. And thank you very much for your attention.
I have a question about RNA families that have variable length stems and loops. How well does aliFreeFold handle those types of families?
Yeah, I think that this go back to the way we compute the feature vectors. Here, typically, we just record all the multi-loop, whatever the variation in terms of sequence length we have here. So even if two suboptimal structures have one multi-loop but of different size, here the score of the weight of the multi-loop, the conservation weight of this multi-loop will be really high. So we consider this fact, but also we consider in the end motif representation the length. So we have both information. We have information about the motif and the information about the variability in terms of length. So this is not really a problem in our case.
Quick question. So in your testing validation phase, so which negative set did you use? Did you try applying this to sequences that are not necessarily from given families but may or may not have a structure? You see whether there is a high rate of false positive or maybe new structures?
We did not test that. We just test on families that we know have a conserved structure. But of course, there are families which do not have any conserved structure because they have they are conserved from the perspective of the sequence. So no, but as we are only focusing on the structure, we just design the experiment with that in mind.
So in your N motifs representation, how did you decide on the N?
Sorry?
How did you decide on the N?
On the end of the N motif?
How many motifs? How many motifs, right? And then if you're recording them like this, if I understand this correctly, you're sort of, and then you're weighting them.
Yeah.
They're considered independently. But
Yes.
I would guess some of them tend to co-occur or, you know, coexist or be antagonizing. So
Yeah,
if you get
correct.
this, you never get this.
Correct.
And it doesn't seem to be playing in your weighting function when you're doing then the averaging. So are you considering it, or?
You mean the combination?
Yeah, combinations for those.
No. We do not consider the combination of those features here yet. In one of my previous work here, when I developed the end motif representation for the purpose of comparison of secondary structure, I add another step to find the combination of motif. But this was done for the purpose of comparing RNA secondary structures.